Now, growing animal viruses in the cell culture. The process by which the cells are grown under the controlled condition in the cells is called cell culture. What are the controlled conditions? The animal viruses can be cultivated in the cells, as we have already studied that animal viruses can be cultivated in the inside the animal body, inside the tissues, or they be cultivated in the they can be grown in the embryonated eggs, or they can be cultivated in the cell culture. So in the cell culture, under the controlled condition, it will be called the cell culture production. We can see animal viruses have been produced with the different cell lines on the petri dish. So what does it mean by the controlled conditions? What are those controlled conditions which are necessary to grow the viruses without any human, without any animal body, without any body, just only the laboratories, we are able to cultivate the virus cells. The control conditions vary with each cell type, which kind of the viruses are there, which kind of the cell culture is there, then the control condition is there. However, there are some special kind of the nutrients which are important and which are needed. Some of the amino acids and the carbohydrates which are essential for the growth of the viruses, they are very important and they should be there. Then some growth factors, there are some hormones which are needed and which should be there in the cell culture so that the viruses can grow rapidly. There are gases which are needed for the life, carbon dioxide and oxygen, and then physiochemical environmental factors are there, just like pH, buffer, osmotic pressures. So these are the basic ingredients, the basic environmental conditions, the control conditions which are important and which should be there in the cell culture. If we are able to produce these conditions, we are successfully can grow the viruses in the cell culture. What is the cell culture? How it started? What is the history? How it originated with the cell culture? How for the first time in the world we were able to know that cells, we are able to grow the cells inside the laboratory. Cell culture was first successfully undertaken by Ross Harrison and it was in about 1907. However, its history was much before than the 1907. There are some special occasions that we were able to know that there are some of the viruses, we are able to grow some of the viruses outside the body of the individuals. Ross in 1885 maintained the embryonic chick cells in a cell culture. However, it was not possibly called cell culture at that time, but he was able to produce them outside. 1878, Bernard proposed physiological systems of an organism, and these physiological organisms were maintained in the living system even after the death, which also provided us the basis for this, that we can be, even after the death, if we produce the similar kind of the physiological conditions, we can be able to see the viruses in the cells. Then again, the Ross, he maintained the embryonic chick cells in the saline culture this time. This time the Ross, who already maintained the chick cells in the cell culture, and now he is in the saline culture, he was able in 1885. Then similarly, the 1897, the Loeb demonstrated survival of the cells isolated from blood and connective tissue in the serum and plasma this time. Then 1903, Joly observed the cell division of the salamander leukocytes in vitro. In the laboratory, producing some kind of special kind of the experiments, Joly was able to know that we can salamander leukocytes can be produced, can be grown in the vitro. Then in 1907, when the cell culture was successfully undertaken by Ross Harrison, cultivated the frog nerve cells. He was able to cultivate them in the lymph node and by hanging drop method and observe the growth of those nerve fibers, nerve cells were in vitro for several weeks. At that moment, we were able to know that we can grow the viruses and considered as the father of the cell culture. We can grow the viruses in the cell culture, and that's why the Harrison is considered as father of the cell culture from the 1907. Why the cell culture and why applications of the cell culture, from where we can use it, the cell culture? Most importantly, for the studying in the bioengineering, in the molecular biology, in many other tools we can use the cell culture. However, the studying the basic cell biology is very important for the cell culture. Interactions between diseases causing agents and cells. Sometimes we use the cell culture to see the effects of some drugs which we have already produced and trigger of the aging, and nutritional studies also we are using the cell culture. Study of the effects of some drugs or some vaccines, we are using the cell culture. Then it is very important for the cancer research. Study the functions of various chemicals, virus and radiation, and to convert the normal culture cells to the cancerous cells, we use the cell culture. Then similarly, in the field of the virology, we are using the cell culture for the cultivation of the viruses to see their different effects. We are using the vaccines. We are using the infectious cycle of some viruses. We are using them in the cell culture. Some of the viruses or some of the diseases in which we are not able to use the animals, or we are very expensive, we cannot use the animal models, we cannot use the tissues, so we are using the cell culture. Genetic engineering, production of the commercial proteins, production of the DNA vaccines, recombinant vaccines, large-scale production of the viruses for use of the vaccine production. For example, the polio, rabies, chickenpox, we are using them as the cell culture as a model. Gene therapy is also very important tool, and we are using it for the cell culture. So two type of the cell culture we can use? One of them is the mammalian and other is the non-mammalian cells.